So she was an undergraduate at the University of Michigan, apparently expecting to transfer into music school, but then ended up in biology and marine ecology. I'm curious about what you play, but that can be a conversation for afterwards. And then she worked in aquaculture research for 3 years and ended up in Israel, which led her towards a PhD working with fish larvae in Scotland. She's focused a lot on larval and juvenile stages of fishes in Lake Michigan, New England, the Irish Sea, North Sea, and the wider North Atlantic, both in the lab and the field. And we're going to hear all about this today. But in 2003, she became a professor in the University of Bergen, and 2018 took a 4-year break to concentrate on teaching and curriculum development at Imperial. And of course, she works with fish otoliths. That's how we know each other. And so I'm hoping there's going to be some otoliths in today's talk, but I'm not sure. Okay, no otoliths. All right, well, I'll stop talking now and leave it over to you. Thank you, Audrey.
Thanks very much. I will start in a minute. I just have to show off my t-shirt, which is the early life history sections of the American Fisheries Society. Early life is good. It's my one time that I get to wear this t-shirt in celebration of fish larvae. So as I'm sharing my screen and getting this all together, I will also tell you about that amazing the traditional joke about larval fish ecologists. I do tend to describe myself as a larval fish ecologist, and then I think twice about it because I think, well, which is the adjective there? And which is the noun? So I do say sometimes that I'm a larval fish ecologist, and when I grow up, I'll be a real grown-up fish ecologist. But don't keep your breath, don't hold your breath for that to happen anyway. So yeah, part of that, of course, this isn't the presenter view. This was just to shock everybody. I don't have 75 slides. I've been a lot more controlled than that. I promise, I promise. Okay, so I call this "Don't Put All Your Eggs in One Basket" because I wanted to emphasize why larval fish, number one, why they're so fascinating, but why it's really, really important to apply the same kind of evolutionary and adaptation thinking perspective to fish larvae that we apply to their parents. And often we don't. We ignore the fact that fish larvae are subject to selective pressures often. We don't consider them to be as diverse as their adults are. And I'm kind of curious as to why. I have to say that I don't have a lot of answers. I have a lot of examples to show you, but I don't really know why this is true. But I do know that fish do maximize their diversity. And that starts right at the beginning. So, I mean, a lot of people, if you say that you work on fish, they say, "Oh, are you a fish doctor? I have my, guppies in my aquarium. Can you tell me about it?" A fish is a fish? No, they're not. Fish are incredibly, incredibly diverse. And they're diverse in a lot of different ways. So if you just take the straight species richness thing, yeah, they're the most diverse of all of the vertebrates. So half of the vertebrates are fish. Yeah, I know. People who work on beetles, yeah, you definitely have the prize there. But in the vertebrate world, fish are the most diverse. But it's not only that they are different species, even within species or between the species, they vary in life histories, they vary in their habitat, they vary in longevity, they vary in growth, they vary in everything. So it's very, very difficult to say what a fish is like, because they are like so many different things. They've adapted to so many different aspects. Why are they so diverse? I'm going to take a time out here just for a second, because it's really important to understand what we mean by fish. What is a fish? So those of you who think phylogenetically, you know that fish is like the most awkward grouping that we could talk about. because actually, if we go back here, what we think of as fish here, if we use the term fish, technically it's a paraphyletic group, right? Because we've got these stupid tetrapods in there. So if we were going to be really technically, fish includes everything, then we would have to be fish. Maybe we're tetrapod fish or non-teleost fish. Obviously, that's not really very helpful, and it's not where I'm going to be talking today. This is, I hope, the only phylogenetic tree that I'm ever going to show you. I'm really going to talk about this group here, the bony fish, Actinopterygia, and in fact, it's going to be mostly all of these teleosts here. And I'm going to call those fish and just be done with it, okay? So any of you evolutionary biologists, don't kill me, or kill me much later, please. We'll go back now. The question of why are fish so diverse? Well, part of it is time, and part of it is the weapon of mass destruction. No, actually, I mean WGD. So fish have been around a really long time, okay, 400 million years at least, right? And at the base of the teleost line, so that's like 350 million years ago, they went through a period, they went through some events, whole genome duplication events. So there was one really at the base of the vertebrates, and then there was a teleost specific event that occurred after that. The main point of this is that whole genome duplication events, they actually build in diversity. They build in genomic diversity. And generally, we consider that those events lead to or set up the conditions for increased adaptation, increased radiation. That's part of the explanation of why, or part of one of the explanations of why there could be so many fish species, why they are so diverse. In some of the groups, oddly enough, in salmonids and in a couple of other groups of freshwater fish, half of those gene pairs are still retained. So a lot of groups after this whole genome duplication, you kind of pare down the number of genes again. But in some of the groups, actually, a lot of those pairs have been kept. And so that is sort of scope for diversity. One of the areas that is really important when we start talking about larval fish is how they got there. So in case your parents didn't tell you, there's this thing called sex, and fish have that, but they do it in a lot of diverse different ways. It's not really very straightforward for fish. Here again, they're more diverse in how they do reproduction, not just in the behavior or not, but just in the fundamental of like, how do they determine sex? So there are two things going on with fish. You've got sex determination, which says whether you're going to be a male or a female. And then you've got sex differentiation, which says whether or not you're going to have functional ovaries or testes. And those are two very, very distinct processes, especially in fish. And fish are diverse in both of those. So in terms of the sex determination, what causes the individual to be a male or a female? Many of the species, well, half of the species, it's under genetic control. So you've got sex chromosomes, just like we do, right? And in most of the cases, in most of those half of the species that do it that way, the heterogametic sex is male, but there are also species where the heterogametic sex is female, the ZWs, like some birds. So, and in other cases, you've got a mixture of sex chromosomes, and you've got sex determining genes on the autosomes, and the expression is really labile. And so there's a lot of scope for diversity, even when you have genetic control over sex determination. There are also species where it's environmental control, or social control, or physiological, in other words, the condition control. So there's a lot of scope for variation in fish for sex determination. Sex differentiation is another layer where you've got this wide scope for diversity. You've got gonochoristic species, in other words, you've got males and females, And that you can have either differentiated ones, so you start out with this immature gonad and it either develops as a male or as a female, or you can have these undifferentiated ones where you've got this immature gonad and it goes through a process of first as a testes and then as an ovary, or first as an ovary and then as a testes. In comparison to those, you've got all of these hermaphrodites. And we generally talk about two different kinds. We've got the protandrous, the ones who are first male and then become female, and progynous, those who are female and then become male. And not all of the species do the same thing here. Some of them it's obligatory. All individuals will first become one and then another. In some of them, some will remain the same sex all the way through. They'll have the same functioning gonad all through their life once they mature the first time. So with all of these things, you already have built in the ability to have a widely differentiated reproductive system. And that gives you a clue that you should end up then with the beginnings of the offspring already being primed for diversity. And that's exactly what happens. I should say that there are some examples that you probably know about in terms of that sex differentiation, because we really like the behavioral responses, the guys that switch sex due to behavioral conditions. And of course, if you all remember Nemo, and Nemo shouldn't have been his dad, he should have been his mom, of course. So we've got those that are male first, and if there's the absence of a big female, then the largest male will switch sex, or when it gets to a certain size, they'll switch sex. So those we know from our friend the anemone fish here, and also the gilt-headed sea bream, which is a commercial and aquaculturally important species, and working with a hermaphroditic species that does this sex change that you want to manage commercially has its own interesting aspects. Most species that are hermaphrodites are actually progynous. They start out as females, and then the largest one becomes male. If you remove that male, the next largest one develops testes and becomes the functional male. So, does any of this fall into some kind of phylogenetic pattern? Can we explain it by, by our ideas of families or groups of fish? Not really. So if we just look at the clupeids, the herrings, for example, here we've got sprat and herring up here. Here we've got shad and menhaden here. Are they similar at the surface? Maybe. But actually, if you look at it, no, they're not. They're quite different in terms of how they reproduce, the behavior, the way their eggs and larvae are distributed, and everything else. So we can't really generalize based on evolutionary patterns. So what's the point here? Well, given that background, it shouldn't be surprising that fish larvae would be diverse, but we don't usually think of it that way. So what I really want to do is focus on some of the different kinds of variation that there are. I hope I'll show you how diverse fish larvae can be just within a small collection. And what happens then? What do we think about that? What kind of variation is there, and what kind of adaptation strategies does that suggest? I'll talk a little bit about egg size and temperature-dependent development rates, metabolic rates, hatching, those kinds of things that have a physiological basis and show you how much variability there is there. And I'm going to use flatfish as a really good example when we want to ask, where is the most important selective pressure? Is it on the larval stages, or is it on the adult stages, or somewhere in between? To me, this is why larval ecology is so fascinating. One day I'll grow up and not be fascinated anymore by larval fish, but in the meantime, I'm very happy to be a larval fish ecologist. So are larvae diverse? Yeah, they are. This is simply from the Bible of identifying the eggs and planktonic stages of British marine fish. And you can see here, these are eggs drawn to scale. And you can see they come in different sizes. They're pretty much the same shape, although there is some variation in shape. And they have different structures. And so they are obviously diverse. If you look at these cartoons, these outlines, sketches of a bunch of quite common species around in our waters, and we don't even have to go into, you know, the more exotic regions, you can see that there are a lot of different forms that larval fish take. And maybe that shouldn't be surprising. I mean, if adults are different, then why aren't the larvae different? Not that the larvae are different in the same way. You can't tell, for example, that by that larval form that it's going to grow up to be something else. But we shouldn't be so surprised to see diversity at the larval stage as well, although most people are because they don't think about it. Here are some direct examples, some direct comparisons. These by Russell are, of course, are done not to scale. They're done, you know, so that they will all fit on the page of his book. But if you look at them to scale, you get another idea of exactly what kind of variation you can see. So here I've collected in these photos 3 species: salmon, halibut, and cod. And here are the eggs of salmon. Here's the egg of a cod, but you can't actually see them here. Here are the eggs of halibut. And you can see that there's an enormous difference in the size of these, just the size. The size at hatching is also really different. This is the size of a salmon at hatching, the size of a halibut, the size of a cod. So, and that has certain implications in terms of what they can do. The other really important thing to notice, if you look at, this is a newly hatched salmon, this is what a newly hatched halibut looks like. These are the newly hatched cod. Here you can see them both in the same dish. This is the halibut. This is the cod. The importance here is that this is, you can see more clearly here, the salmon, when it hatches, is actually pretty well endowed in terms of its organs. For sense, it's got a functioning mouth, it's got a vascular system, it has muscles, the whole shebang. As you can tell here, this halibut is pretty bereft of functioning organs. It's very transparent, and it can't do a whole lot. It has this huge yolk sac, but then so does the salmon here. Just from looking at this, you can see that there's this incredible range of capabilities that these organisms have when they get out into, as free living organisms, free living embryos, out into the environment. A lot of people consider these, even though they've hatched, to be embryos. And in fact, that's something that it's important to look at here. Sometimes we can see that these guys hatch at different stages of development. Again, the salmon up here, you can see the fish in it, or at least I can. This is a plaice, flatfish. Its eyes are pigmented, which means they can function. It also has a mouth, and it has a complete alimentary tract, so it can feed. This is a hake larvae at hatching. The eyes are not pigmented, so they're not actually functioning. It has no mouth. It has no alimentary tract. It can't really do anything. It does have muscles, so it can twitch. It does have neurosys, so it can actually sense the environment. But in terms of the capabilities of these three at the same stage of development, as i.e. at hatching, they have hugely different competences. And of course, their interactions with their environment and the adaptations and the strategies that they might have are going to be fundamentally different. So if you just work on one of them, then you're very, very used to seeing things only from the view of that one species and that one, let's just say, model of a larvae. If you work on a number of them, then you actually get to see what one species can do and one species can't do, and start that comparisons. There aren't a whole lot of people who work on a lot of different species. We joke in Norway that we're multi-species. We work on cod and herring, but it is actually difficult sometimes to work on a lot of different species. You want to focus on the one and, you know, see it through in terms of the developing a line of research and figuring out the questions about that one species. But the comparative approach actually has a lot going for it, because it allows you to appreciate just how many adaptations we see in these things that don't seem to be able to do anything. Just taking the herring example, well, actually the clupid example. Remember before I talked about whether or not the species makes any different, or whether or not we've got any familiarity, whether we've got any similarities that we can hang on sort of the phylogenetic level. Here is one of those situations where here's an example of why we can't. So in the clupids, we have herring, we have sprat, we have shad as well. The herring are great. Herring actually have these amazing eggs, which look like typical marine eggs, but they are benthic. They are adhesive and they're stuck to boulders or to seaweed and things like that. On the other hand, the sprat, and if you hold a young sprat and a young herring together, say a 1 or a 2-year-old fish, it's actually a little bit difficult to tell the difference between them. Sprat, on the other hand, are pelagic spawners. These are their eggs, and they float in the water column. This is an example of what they look like. These aren't sprat eggs, these are saithe eggs, but it was the only picture I had of floating eggs to show you. So here we have fairly closely related species, yet they have entirely different strategies in terms of depositing their eggs. And also that means that their larvae, when they first hatch and when they have their, you know, in their first days, have completely different environmental experiences. So one of the ways that we kind of understand that is to think about some kind of gradient in terms of what a fish larvae can do or can't do in terms of its development. Eugene Ballon was someone who worked on this quite a lot. First of all, he started out by defining ontogenetic stages in fish, and then he tried to think much more broadly, trying to link the development of fish to reproductive mode and to try and link both of these to evolutionary patterns, some of which made sense and some of it didn't make sense. He gave us some amazing terminology and concepts. He called larvae free-living embryos or eleutherembryos. He had a whole system of terminology for the different stages that many of us kind of put to the side because we couldn't pronounce it. He gave us also this concept of precocial and altricial. So altricial meaning that you've got almost direct ontogeny, where the larvae are very well endowed in terms of quite well developed when they hatch, and they don't go through any major metamorphosis. Precocial being those species where what hatches out of the egg is really still quite an embryo and goes through a much more prolonged development with a very, very clear metamorphosis in it. And he called that either indirect ontogeny, and that they were precocial. They were hatching too early, you could say. So my question has always been, what's the typical marine fish larvae really like? Is this is hake here. They grow up to look like this. What is actually typical? Is this a typical marine larvae? I don't know. It's a baby fish, but is it really very much like a fish? Maybe not. Most fish larvae are plankton, most fish larvae. Certainly our typical small egg, small larvae, they're not actually acting as fish for the first part of their life. And it could be as long as 3 months or 6 months for some of them. So they really are more like a member of the plankton. Does that change how we think about them? I don't know. Maybe it could, or it should. Quite a while ago, Weinmiller and Rose produced this sort of thought piece, this modeling piece, and asked that question: why do most fish produce so many tiny offspring? Why is that a successful strategy to, spread these embryos out? And they actually modeled fish larvae of different sizes in different prey environments that were clumped and prey environments that were where the prey was randomly distributed. And they looked at cohorts of the same biomass, but divided into either fewer large individuals versus many small individuals. And in fact, in low prey conditions, this measure of contagion tells you how patchy the environment is. This is hugely patchy here. If the conditions, the feeding conditions, the average feeding conditions are not great, then large larvae always win. However, if you've got conditions that are quite patchy, reasonably patchy, and in those patches you have high densities of prey, you do much better if you have lots of small fish larvae. Okay, all right, so you can win both ways. That lets us know why there are still so many broadcast spawners around. Fine, but larvae don't stay in the plankton. They move from being these like drifting things that are just trying not to get noticed and eaten by something else, through to different stages where they become more and more capable, until eventually they end up looking like little fish and are in fact the top predators or settle out of the plankton entirely. I'm not going to spend a whole lot of time talking about this. I can spend hours talking about it afterwards if you want to, but I'm going to go on now because I'm conscious of the time passing. This is a hake, by the way, and you can see these guys are like 2.5 millimeters at hatching. That's really tiny to be out there. I mean, you've got copepods that are, 1 millimeter, 1.5 millimeters. So they're in the same level of a lot of our favorite plankton. But okay, we've got a reason why there might be lots of these free-living embryos around. Is it really such a crazy strategy? What kind of adaptations do these guys show? What is their versatility? These are safe in the background here. This is a lemon sole. How come we've got these different things, and what can they do? So I wanted to show you something about these flatfish, because here, this lemon sole, it spends a lot of its time as a larvae like these would do, but actually it has to get from here to here. Flatfish are even more crazy when you talk about the strategy of having a larvae. Why would you have, if you're going to end up asymmetrical, why would you start out symmetrical? And how do you deal with the cost involved in that whole metamorphosis thingy here? I think I worked on, I worked on place during my PhD, ages and ages ago, and a couple of other species as well. And dealing with flatfish really starts you thinking about why would they even do it that way? I mean, that's absolutely crazy. I did want to show you what it is actually like. And I don't know if you remember your teenage years or if you have teenagers at home, going through this kind of a change, going through this kind of a rewiring is really traumatic. So if you think of these poor guys, one of the things that people notice first is the fact that they end up being flat, asymmetrical, but watch the eye. That eye migrates, and it migrates through torsion in the skull. So that means during a portion of time, and we know this from experiments, the information coming from that eye is at best unreliable and worse, not at all. So you've got something wandering around in the pelagic region trying to feed with one eye not functional. And it's by design. No, I don't mean by design, but I mean, it is the way that they're supposed to be working. So how can that be adaptive? Well, obviously, there are the advantages to being in the plankton. There are advantages to eventually settling out. And if you're going to be a flatfish, you are going to end up on the bottom. So you have to make that transition in some ways. With many fish, with many of the species, it's actually done offshore, where to a certain extent, some of the predators that these small guys would be susceptible to aren't there, bird predators and other invertebrate predators, we think. There are some other areas where within a species, you can end up with unsuspecting adaptations, or rather adaptations that we haven't realized are part of adaptations. I'm going to show you some things here that have to do with eggs and temperature and spreading your eggs across a lot of different baskets. So this is information about place again, but about the reproduction. So this focuses on where you put your embryos. One of the very interesting things that happens with fish that are Hydroparis that spawn several times in a season, they spawn in many, many batches throughout a spawning season. As you can see here, the time is going on. There's actually a decrease in egg diameter, especially within a single female. The egg diameter, or in other words, the size of the egg, tends to decrease over time. One of the other really interesting things that we've only realized recently is that at the same temperature, the development rate of those eggs changes. So you've got two different things going on. You've got larger eggs being produced earlier in a season, smaller eggs later, but then some kind of probably physical control of development rate that isn't equal, isn't constant across egg sizes. So at the same temperature, eggs of different size will develop at different rates. And this can help actually to spread out the hatching and the coverage of the spring bloom, for example, pre, mid, post, not only with the egg size, but also the fact that those eggs respond differently to temperature in terms of their development. Pretty cool. We've also got hatching itself. So at what point should I actually get out of this egg where I'm fairly safe and out into the environment? So hatching isn't a developmental event. It's an event, but it's not a developmental stage. Here's an example from herring. This is a newly hatched herring. These are newly hatched herrings. These are herring that are the same age from fertilization as these guys here, but these were still in the eggs. I opened the eggs and took out these herring, and really physically, they're not a lot different. They're a bit more curled because they've been in the egg and they haven't had time to fully stretch out. But there's not a whole lot of difference between them, except that one was in an egg and the other was already swimming. If we look at them, we can actually see here that even though these open ones are the size of larvae when after they were released from their eggs, these are the solid ones are ones that hatched on their own, for example. And even though the ones that I released from the eggs were smaller in the beginning, by about 3 or 4 days, they're all the same size again. So here's how they look in their egg. Here's how they look when they just come out of the egg. You know, it's not such a big deal. They all catch up. So when to hatch, maybe that's another arena where you can actually spread the risk across a number of different baskets. Herring's also incredibly interesting in that at the metabolic level, they seem to be able to do their own thing no matter what the temperature is. So we know that herring have a lot of small, locally adapted populations. They experience a range of temperatures. If you put them in the lab and you test them across a wide range of temperatures, we can find that metabolically, they're doing pretty much the same thing. This is at hatching. This is at the end of their yolk sac stage. Fitness of the yolk sac larvae and the larvae when they've finished their yolk sac is approximately equal across this whole range of temperatures. That's a pretty neat feat. And it means, of course, that not only can you adapt to local conditions very easily, but also if there's any shift in those conditions at the physiological level, you're not at a disadvantage. You're not at a disadvantage. They just keep on doing their own thing. I just wanted to show you again some of the other types of diversity that we've got in fish larvae. I thought I'd take tuna here, about 200 kilogram things. They've got these incredibly small eggs, hatch at about 3 millimeters. This is what they look like after 20 days. Super fast growers. They develop really fast and they become predators and piscivores at an incredibly early age. So they've got that ability to switch very, very early. And that, you know, compared to the place or the other things that we saw, you can see that immense diversity in what a fish larvae is. So we've got differences in buoyancy, we've got differences in where the larvae are. One of the questions that I have is what part of the larvae is under adaptation? Of course, it's really, really easy to say everything because, well, you know, we've all been marking essays where the conclusion says it could be this or that, and it's probably both. But I am going to say it could be this or that, but it's probably both. But one of the things that we're not doing enough of, I wanted to say, is when we talk about fish larvae and we look at what fish larvae can do, we don't have enough evolutionary thinking behind it. We don't have enough of thinking of what are the adaptations. And part of that is because we don't collect enough information on how widely they vary, even within a species and even within the batch of a single female. I'm going to finish here. I'm going to say thanks, and I'll be really happy to talk to anybody about any questions they have.
Thank you so much. We're clapping. That was really interesting, really interesting. It's funny, me and Leanne were texting me that I wonder if she'll do a guest lecture for our marine vertebrates course. So,
Sure.
Excellent.
I won't wear this though.
Brilliant. If anyone has any questions, please add it to the chat and let me know if you're happy to be added to talking, on video. I have a quick one to get going. I'm just curious about the herring. We've just been talking to my master's student about herring. We're just talking about the population crash and everything. And I was like, when he said that they've got this specific eggs need to stick to substrates, I'm like, oh, do you think that had any factor in the crash, these kind of specific egg needs, or I don't know, pollutants, or or was it just they were overfished?
I think they were probably overfished. It's very interesting for the herring because, as far as we know, except for, why is this not? Except for some of the North Sea populations, there hasn't been a lot of problem in terms of the spawning areas. That's not true in the Baltic, where hypoxia is a big problem. It's not true in Pacific herring, which actually spawn almost intertidally in the, in littoral or in the sublittoral zone where there have been oil spills and things like that. In Prince William Sound, King William Sound, in Alaska, where there is the big oil spill, that impacted a lot of the spawning areas. But otherwise, they can spawn on these big boulders or in cobbly areas, and I think that's a much more local effect if there's a problem in that specific area. Gravel extraction in some parts of the North Sea has been sort of.
Well, we just had our Greenpeace guy, the one who was dropping the boulders a couple of weeks ago. So, just helping the herring, Sorry. No, that's interesting. Has anyone else got any questions? I'm really unfamiliar with this webinar format. So I don't know how to find out if anyone has any questions. Oh, here we go. Tom Cameron, brilliant. So let's add you to as a panelist. That'll be fun. Tom's always got good things to say. So allow to talk.
Yep, I think I can talk now.
Yeah.
Thank you.
Hello.
Hi there. How are you doing? Thank you very much for that. That was brilliant. I wanted to ask about having all the strategies in one basket. And so you could think of, I could hear the different mechanisms you talked about, mechanisms across the populations and the diversity across the populations. So you can imagine, but you can also have the idea of diversity within females. So, what evidence do you think is that all species are bet hedging, or is there an aging process where they tend to produce one strategy when they're in their best condition, and different strategies at different parts of their lifestyle, or even within a breeding season? So some eggs within the produced from the best quality fat are better eggs or whatever. I mean, is the diversity across different types of axes, is that the right word?
Yeah, it's certainly the traditional view is that within a particular spawning batch, if we're talking about our, let's be Eurocentric here. Let's be Northwest or Northeast Atlantic here. If we're talking about our typical marine species that we know a lot about, then we have the most diverse, we have the most diversity between females, especially or in the eggs produced by different parents, especially of different sizes or ages. And then within a particular set of parents, you would have less diversity. And then within the single batch of two parents, you would have even less. Of course, we do have diversity at all of those different levels. And yeah, as I was showing with the place eggs, that the size of egg changes over time. That could just be a mechanism of declining. I mean, it's always been explained by declining condition of a female over the season. But it does have that knock-on effect. You might ask then, well, rather than suffering that decline, why not just put more eggs into fewer baskets, you know, more eggs at each spawning event? And why doesn't a single female then hone in on just one event? There has to be an advantage to spreading that. But not only that, because the eggs are of different sizes and they develop at different rates, even at the same temperature, then you increase even more that spreading. So it does go on at all of those different levels. I honestly couldn't tell you what a cleaner wrasse does, because I've never studied it. And that's part of our problem. Our view is of course honed by these very well-studied species. You know, we don't even actually include in our thinking all the stuff we know from zebrafish development. That tells you how bad we are in terms of looking truly at what's going on during the early stages of fish. We know something about salmon, but not as much as we should, probably. Not out in the wild. We know much of it from hatcheries. I think that everything that is, all the pressures that are saying this is where it's successful are pushed away again by those pressures that are saying, yes, spread it here, because the environment, especially for the highly fecund species, they're still trying to hedge their bets across the varying environment.
I guess that makes sense. I mean, part of the reason I asked that is I did some work on an invertebrate model system, but it's actually a very fish-like life history where adult duration is longer than juvenile duration. Fecundity is linked to female body size and things like that. But and they have very much a within female system where, you know, when they're immediately 3 days after they're sexually mature and nice and big and plump and full of all the best resources, they're producing very relatively few but very large, well-provisioned eggs. And it's almost as if they are just trying to get the most out of their body as they age, that they then seem to switch into a produce lots of small eggs. And I think sometimes we look at female strategies across species, we think, well, those females produce lots of eggs. They're clearly they've just got a strategy of producing lots of eggs. But within that strategy, again, and I can't work out if that is hedging their bets so that the offspring meet stochastic environments, some win and some don't, or if it is just, what's the best thing to do now that I've only got 50% of my body condition, and there's actually selection of strategies at different stages. It's difficult to describe, but it's certainly a fascinating topic.
It ends up also getting tied in with the oocyte development, the gonad development itself. I mean, a lot of the fish can't switch once fecundity or realized fecundity is determined, say a month or two before spawning, then you have to go with that. The only thing that you can do is not spawn those, or if you run out of yolk, then, or you run out of energy, then you can't put as much into those later ones as before. So, but there are species that are spawning every day and are in fact going through that last stages of vitellogenesis on a daily basis, and they should be able to be much more fine-tuned to what they're putting into their eggs. So I think they could do.
I just wanted to jump in with a mini parallel there, just because I thought it was really interesting. With the salmon work, we're finding that with some of those really slowing growing individuals, the juveniles, are spending like an extra year in fresh water. And this is in the US where it's really hot. So that's pretty rare strategies still around in this particular region. But I just thought it was quite funny because there's just so much obsession about we've got to maximize growth rates in terms of management and restoration, and we've got to get them out big and early to the ocean. But actually in some years, those ones that stuck around, the slow growing ones, were like the winners that came back to spawn. I just think it's kind of funny that we can sometimes try and design things in the best possible way, but reduce that diversity that's so important and what they've evolved with. So I just thought it was interesting with the egg changing size. Anyway, I'm not going to hold the fort anymore because we've got some more questions. Oh, Tom, did you want to add anything before I?
No, all done, thanks.
Okay, thanks. Com Denishot, is that how you say your name?
Yeah, Com, yeah.
Dr. Denishot, as of yesterday, right? Congratulations.
Hi again, Audrey.
Hi.
Thank you. That was super interesting. I had one question, which is because I don't know anything on fish larvae, but I remember in my master's degree we had one lecture on fish larvae, and it was on Réunion Island. So we had this one scientist who was doing basically all of the fish larvae work for the entire Indian Ocean. And one thing that he was stressing was that it was very understudied, and only a handful of experts that knew how to properly identify and study these fish larvae. So I was wondering how much, I mean, is there a potential issue where we don't know much about fish larvae because we cannot really identify them species by species outside of like just the very, very known species? But is there like a lack of knowledge on what they actually look like and what they actually do?
It's like a lot of other things. It depends where you are. So in some areas we have really good knowledge of what the species are, and the descriptions are really good, and we've got great collections in different places. And despite that drop in taxonomists, there is a good management reason to have people be able to identify fish larvae. And so the number of people who can actually identify different species of larvae, especially the commercially important species, has been increasing in recent years. So that's true for a lot of the North Atlantic, but it's less for the non-commercial species. If you go to some place like the South Pacific or parts of the Indian Ocean, that's not the case. We probably can do families, or we can do orders because they look like what they should be. But there are some areas where the identification is very poor. The flatfish larvae of Australia are not completely documented. And there are a couple of people who have had that as their mission for the past 2 decades to do. So yeah, it depends where you are.
Okay, thanks.
And do we have a question from
Yanni?
Let me just make him allowed to talk. There we go. There you go. Take it away, Jochen. Is it? Sorry, it might be.
Can you hear me?
Yeah, there you go. Yeah.
Okay. So should I switch on?
Yeah, it'd be nice to see.
Can you see me now?
I think it's
still not.
Maybe not. We can hear you though.
Okay. Okay. I'm not a specialist at all in this fish larvae. And it's quite funny. I actually found out about this presentation through the Twitter of Combe, who asked the previous question, and I saw his PhD, which was really nice as well. And so I'm very glad that I could get this introduction on fish larvae. It was really interesting, and I wondered. Now I'm moving into this world of stock assessments, and of course the stock recruitment relationships are kind of like the holy grail. If you know a bit how to predict the recruitment, and I was wondering, as we have this upcoming project where we want to investigate effects of climate change on recruitment, what kind of advice you would give us? It is maybe not a really fair question, but well, I have asked now.
Okay, where are you?
I'm in Belgium, and we work on place and soil in the Northeast.
I don't
so
know.
the North Sea, English Channel, Irish Sea.
Okay. It is actually an area that's very intensely being looked at, both in IC's working groups and also in EU projects. What is the effect, or what are the potential consequences at the recruitment level for climate change. I mean, what happens if species change their distributions and they're spawning in different places, or what happens if, well, ocean acidification, of course, has been investigated by scientists, especially Katrina Clemmensen from Kiel has looked at it quite a lot. And just changes in temperature, both at the, you know, for all of those species. And I can give you the names of some of the people, somebody that you could contact. In fact, the best person to contact might be Myron Peck, who has just taken over as head of NEOS.
Yes.
That's his thing.
Okay, I'll try that.
I mean, the real question will be whether any of these species get pushed beyond what's going on, or is there going to be another flip in terms of the food sources available? What happens if you flip, you know, between Helgolandicus and Finnmarkicus in terms of the North Atlantic, the two major cavanoids? Or what happens if you've got species that are completely not reliant on any of the cavernous, but can do quite fine? Will they take over? Those sorts of questions. What happens if we don't have a winter? We don't know anything about overwintering larvae.
Mm-hmm.
Maybe we will start having more of them because they'll survive.
Or maybe there will be less. For sole, I know they say if they're very cold winters that they
do better.
Yes.
Yeah. And for place as well. But that has more to do probably with the recruitment of crangon and predation on the nursery grounds than it does actually during the pelagic stages. That we think that's the sort of the explanation.
Crangon eat the larvae, or the other way around, the larvae eat the crangon. Sorry.
The crangon will eat the very first settling larvae. So if it's cold, then the crangon are delayed. And so more of those newly settled larvae actually survive. That's the accepted explanation. It doesn't, but it is true that cold years tend to have very, very good survival off the nursery grounds, not necessarily recruitment to the fishery, but survival on the nursery grounds.
Those multi-species interactions seem to be so, I mean, they're so important, but they're so hard to extrapolate. Yeah. Cool.
Thank you very much.
Thank you. Thanks for coming and for asking that question.
It was really interesting for me to have this. Yeah.
Thank you.
Yeah, thanks for joining.
World open.
Great.
Thank you, Com, for spreading the word.
Yeah, that's awesome. Yeah, so I think anyone, I can't see any other hands up, but I would say just unmute yourself. I don't think you're allowed to on this new format that I'm so unused to using. Okay, so I think we've got all the questions. Some people have said thank you, and Michelle Taylor was talking about the parallels of deep sea coral larval strategies, and yeah, that's been a really fantastic talk. Thank you.
Thank you very much. Thanks for the invitation. I really, really enjoyed it. And I love to be able to talk about, I did have a picture of an otolith back in the...
Next time, I'll have to get you back.
Next time.
Oh, well, yeah,
thank you very much.
And we'll definitely be asking you to come back for our fish, marine vertebrates lecture series. So thank you. Okay, thanks everyone for listening. Okay, bye Audrey. Bye everyone.
Bye.
Bye.